This program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest UCTV programs.
Looking forward to this opportunity to talk about especially some of the newer work that we've been doing in my lab in collaboration with others that I'll be talking about, and then especially to get some feedback and listen to some of the questions that you have after my talk. And so thank you all very much for coming and be willing to listen to me for the next 45, 50 minutes or so. Okay, so let me begin by talking about the outline. So this is an ambitious topic. I'm going to talk a little bit about language and communication in autism spectrum disorder, discuss the heterogeneity that we know exists, focus in a little bit on subtypes, as well as the neural bases of language in ASD and siblings. That's actually all by background of what do we know about the endpoint of language in this very complex disorder. I'm then going to switch to thinking about what's really in the title. Well, what are the origins? And I always say about my career, I started at one end of the life cycle, focusing on the end point of language, and I've been able to move my way back in time as new paradigms and new tools have become, and new opportunities for collaboration, I should say, have become available. I'm going to talk about then, for the rest of the talk, the work that we've been doing in our high-risk or infant sibling study, focusing in on the development of speech perception, asymmetry in the neural response to language, language processing in high-risk infants, and looking at contributions, that should really say, to language development, looking at both infant and mother contributions, and end by discussing our future directions. Okay. So let me talk about language in autism, okay? The DSM-5, in their wisdom, and I mean that in a very positive way, I absolutely applaud what they have done, have basically taken language out as a core impairment. Having said that, we could either pack up and go home, because that's what I'm going to talk about today, or say, well, even though it is clearly not a core symptom, there's something very, very important, fundamental about trying to understand the phenomenon of language in autism spectrum disorder. Why? Well, first of all, we've known for a very long time, from the earliest studies, the earliest descriptive studies of autism, that it's really the single best predictor of long-term adaptive outcomes, executive functions, social cognition, and atypical behaviors. Children and adults with autism who have better language also have better outcomes, better executive functions, better social cognition, and fewer atypical behaviors. So it's really the most important predictor over and above IQ itself. Effective behavioral treatments, particularly the ones we know that are the most important, ESDM, Early Start Denver Model, ABA, have the most significant impact on language. What they're able to do best in early intervention is to change the trajectory of language development and really show very rapid gains in this domain. For parents, the child's ability to speak is really the most important factor. That's what they're looking for. If their child can communicate with them, especially using spoken language, that really makes a difference. They feel that they can cope better. They feel that impacts on their own well-being. The genetic liability for autism includes language impairment, and in fact, genetic studies that have basically subtyped their sample on the basis of the certain language phenotypes, such as age of first words or age of first phrases, have actually been the most successful in identifying some risk genes for autism than when they haven't subtyped on language. So language is basically terribly important. So yes, we're here today to talk about a comorbid aspect of autism, not a core symptom, but it's arguably, I think, as important as the core symptoms themselves. So just by way of background, we began a program of research about 15 years ago, and one of the things that we did was to look at some of the variability in language. We've known for a long time that, in fact, the universal impairments that we do see that are still a core aspect of the communication piece of the social communication domain of impairment are in the pragmatic and discourse aspects of language, okay? So individuals across the spectrum have problems with narrative, problems with conversation. And this is part of the core impairment. And we have done quite a bit of research, and so have other people, to suggest that this aspect of social communicative impairment is related to aspects of theory of mind, including some of the precursors to theory of mind development. I'm instead focusing on the structural language aspects, and here is where we see enormous heterogeneity. About 15 to 25%, we really don't have a good handle on this right now, are what we call the minimally verbal. They have almost no spoken language skills. The largest group make up the verbal. They do acquire some spoken language, but they remain behind their peers. So they still retain impairments in language relative to their peers. And this is the group that I refer to in my talk today and in some of the papers that we write as the ALI, which stands for Autism Language Impaired. But then there's a group of individuals, maybe 25%. I think this is a group that's growing relative to the rest of the population, who have intact language. So they have good articulation, intact phonological processing, and they're at their age. They're matched to their peers in terms of their vocabulary and their grammatical abilities. This is the group that we call ALN, Autism Language, but Language Normal, meaning that they have intact structural language skills, okay? And this is some early work we had done on these different subtypes, the ALN versus the ALI group, that I had done with a postdoc at that time, Margaret Kjelgaard. We published this work in 2001. She then left the field for a research field for a decade, focused her work as a speech-language clinician, and has recently come back to research, and we're now coming back to collaborate on a number of projects together. And basically what you see here is, on the left, this is the ALN group. We give them a bunch of standardized language tests that tap syntax and semantics, or grammar, meaning, nonsense word repetition, which is a phonological processing skill, vocabulary, and articulation. And you see these kids basically have a flat profile on tests that the average child, typical child, scores 100 on, and that's what this ALN group scores. So that's the definition. They have a flat profile. Their language, structural language skills on tests like this are at the average for their age. But this is the ALI group. Okay, and this is actually the data that come from the children that have IQ scores above 70. And what you see is that Actually, they are at age level. They're between 5 and 14, and their articulation skills, their ability to articulate speech sounds in words, pronounce a B, pronounce the vowel sounds, etc., is actually quite okay. Okay, they are somewhat impaired in non-word repetition, phonological processing, and vocabulary, and they show the most pronounced impairments in higher level syntax and semantics. And this is an intriguing and distinctive profile. It's what defines them as language impaired. And what we proposed in that original paper was perhaps this group of children with SLI are similar to children who have a different neurodevelopmental disorder, specific language impairment. Because when we look at kids with SLI who don't have autism, they don't have frank neurological damage, they don't have cognitive, nonverbal cognitive impairments, they're not hearing impaired, and they don't come from extreme deprived environments. Nevertheless, they have great difficulty acquiring language, they're delayed in the onset, and they show, when you give them at the age of 5 or 6, the kind of standardized language tests that we gave our children with autism, they show that same profile across that uneven profile. So we said, oh, maybe kids with SALI are like children with SLI. So there are 2 clinical diagnostic markers for SLI, for English-speaking children. One is problems in non-word repetition, where they listen to nonsense words and they have to just repeat them back. And also the marking of tense, where if you say, so here's a doctor, what does he do? Well, a child with SLI will say, he study, or he make children cry. Naturally, that's what a lot of our children said about doctors. So for the doctors in the room, that's what you should know children are saying. Well, what do they mean by that? What's wrong there is that they've forgotten to put on the word the S, the makes children cry. Anyway, we gave these kind of experimental tasks to children with autism, and we found that the children with ALI showed the same profile of difficulty on tasks that tapped non-word repetition and tense marking. And it was the same pattern of performance, saying that they weren't as good, their correct responses were lower. We also found that they showed the same kind of error patterns. So we argued that ALI, children with autism and language impairment, which remember makes up the majority of the population, essentially have autism plus a comorbid SLI. We then went on to ask the question, well, what about their brains? Their brain organization for language. And so we've been looking in particular, we know that in the cortex, there are 2 main areas that are then connected by some important long-range fibers. In the frontal area, what's called Broca's area, in the frontal regions of the brain, and then in the posterior area, Wernicke's area, in which the planum temporale is a central feature. And in our structural studies, then, what we're doing is asking the question, well, what's the volume of these areas? Are they the same size, or are they smaller, or are they larger? And also, what about asymmetry? In healthy people, the vast majority, over 95%, even left-handers, the majority of left-handers, these structures are larger in the left hemisphere than they are in the right. So we have this asymmetry. Okay, and then functionally, although in fact these regions are important in processing language in both the left and the right hemisphere, there is a privilege to processing language in the left hemisphere in these particular regions. Well, let me talk a little bit about the structural studies that we have done. So originally, the first study that came out a decade ago by Martha Herbert and colleagues, and then we followed this up in a new sample. What we found was that there's reduced left hemisphere or even reversed asymmetry in the frontal areas of the brain, okay? And I'll discuss that in relation to this graph in a minute. But we also found that there was exaggerated left hemisphere asymmetry in the more posterior, that planum, that orange area on the previous graph, the planum temporale. So what we did, this is the data from the follow-up study by DeFasi et al. But we actually had 4 groups here, and so did Martha Herbert, who actually had children with SLI in her original sample, who she was comparing to children with autism. And here what you're seeing in red are the typically developing controls. The purple are the ALN group who don't have structural impairments. The green are the children with autism but with language impairment, and the blue are the SLI children. And what you see here is this overall group reversed asymmetry in Broca's area. It was a quite extreme sample in this study, and a smaller sample. In a larger sample, we usually just see reduced asymmetry, and that's been replicated in other labs. But we saw this exaggerated left hemisphere asymmetry in the posterior regions, and that we've replicated that a few times in our lab. So we saw that the neural organization, or at least the structural piece, in children with ALI, in particular, in autism with language impairments, was traveling along with the kinds of atypical patterns that we saw in children with SLI. Now, with my doctoral student at the time, Kristen Lindgren, we followed this up. We've been doing a number of different studies looking at the anatomy of language regions, but what Kristen added into this was looking at the unaffected siblings of the children with autism spectrum disorder. So we now were expanding beyond the children with autism to ask the question, well, is this something that maybe travels in the family, okay, some of these patterns, that's atypical, but doesn't actually result in the syndrome itself. And that's something that we refer to as an endophenotype. And finding endophenotypes, i.e. Markers of a disorder that travel in the family but don't necessarily result in the disorder itself, can be extremely important and useful for helping to identify the underlying etiology, particularly teasing apart some of the genetic risk factors for the disorder from additional contributions to it, either additional risk genes or non-genetic risk markers. So in her work, what we did was we had a group of boys with autism, typical controls, and siblings. And here's the data on the volume of the language regions. These are the frontal areas, and these are the posterior regions. The controls here are in blue, and the kids with autism are in red, and the siblings are in green. And what you can see is, instead of traveling with the controls, who in fact are what they're like behaviorally, the siblings, in terms of the volume of these areas, are traveling with their autistic siblings, okay? And then in terms of the asymmetry, we actually found that the frontal asymmetry doesn't travel along as an endophenotype, so presumably those regions are a little smaller in both the left and the right hemisphere, okay? But we can see in terms of the exaggerated asymmetry in the posterior region, we're seeing that they're somewhat intermediate, which is often the way endophenotypes will work out. In other words, this group probably breaks down into 2 groups, one of which actually looks exactly like the controls, the other which looks a little bit more like the siblings. So that's by way of saying we see then that autism has this interesting atypical brain organization, and it's something that's traveling in the families. So now let me move into the second, and perhaps that was by way of background to what we're going to see in terms of the origins in infancy. Okay, now I'm sure everyone here is quite familiar with the notion of studying at this stage infant siblings. It's actually hard to remember that, in fact, the first paper that was published on infant siblings only came out 7 years ago in December 2005 by the Canadian group Swagenbaum and his colleagues. I think we all think that this has been going on for a very long time, but it truly hasn't. And I think this has been a very interesting and very rich area of research. So the idea here is to, there's lots of reasons for studying infant siblings, but I think we all began by asking the question, how early can we see risk markers for autism before the onset of the full-blown disorder? And we want to distinguish in our work between those things which in our group we call the positive risk signs. And those are signs that show a high degree of sensitivity and specificity for diagnosis. And whereas we all thought maybe we'd find some very obvious positive risk signs very early in the first year of life, especially of those of us who thought, oh, well, we're experts, we're bound to see it. I think we know most particularly, I think that's what's been so important about the your projects going on here. That really has been a much harder thing to find. But we want to distinguish them from the endophenotypes. We're all studying high-risk infants, okay? So they're infants that are siblings, just like the siblings in the study I just mentioned. And we're interested then in what do we see that may be different in these infants, but may not necessarily be predicting to diagnosis. And I think distinguishing between that is a crucial part of what all of us in this world of the infant SIB research are trying to do. We're very interested in early developmental trajectories prior to diagnosis. And in the work that we do, and most of the other teams, we're comparing high risk and low risk infants. In our work, we're careful always to control for birth order. So we don't have low-risk controls who are firstborn children. They're always second-born children as well, or third-born, but who have a much cleaner family history, if you will. At this point, we're expecting 15 to 20%, closer to 20% of our infants to go on to have ASD or other neurodevelopmental disorders based on the estimates of other studies. This is an overview, and for me to say that this work is an complete collaboration with Chuck Nelson from Children's Hospital Boston, and we actually carry out all the work at Children's. And it's been a fantastic relationship because Chuck and I bring completely complementary expertise to this enterprise. He was a world-class infant researcher and developmental cognitive neuroscientist, and I brought the autism piece to the project. So it's longitudinal. We see our kids in the lab many times. We do lots of behavioral measures. We're doing experimental eye tracking for both faces. He's also an expert on the development of face processing, and I'm the language piece. So you can see in everything we do, there's faces and there's language. And we're using a number of different experimental methodologies. We do eye tracking, we do electrophysiology, and we're doing now some functional NIRS work. And we also, between 6 and 18 months, we send the families home. Actually, we do a home visit with this, with cameras, and we ask them to send us home video diaries, and we also have online written diaries between 6 and 18 months. Okay, and I've highlighted in red the parts of the project that I'm going to talk about today. And what's wonderful about collaborating with Chuck is that you could bring him here and he could do a complete talk on his own on all the other parts of the project itself. This is just a project, this is a slide to show you how overwhelming this all is, because this is what we're doing with the project at all times. Don't bother trying to read it. Okay, so let me begin by talking about the origins of this atypical brain organization for language. So we talked about how studies of older children and also adults show this atypical brain asymmetry for structural and functional organization for language. I didn't talk about the functional piece, but there is plenty of data on that too. Well, not plenty, but a couple of good studies. And as I said, the structural differences, we don't know about functional differences in older siblings because no one's done that work. And the question is, how early can we see this in infants? To what extent is it an endophenotype or something like a positive risk sign? And so what we're using to tap this is electrophysiology. We're doing event-related potentials. That's what ERP stands for. Looking at that as well as the EEG signal itself to speech sounds during the first year. I'm going to start by talking about the work that's been spearheaded by my doctoral student, Annie Siri. So what we do is we're basing our work on a paradigm that had been developed by Patricia Kuhl. And it's a double oddball procedure. Don't worry about that. Basically, the infant is sitting on the mom's lap. We do this at 6, 9, and 12 months, okay, only. And the baby sits on the mom's lap, and they're watching somebody blow bubbles. That's just to keep them as still as possible. The mom's told to try and keep the baby still, because when you're collecting that EEG, you can't have too much movement going on. It's a little forgiving, but not a lot forgiving. And they're hearing sounds over the speaker, da, da, da. 80% of the sounds they hear are da. 10% of the time they hear what we call the non-native contrast, okay? And that's a Hindi da, okay? I'm told I say it quite well, but I'm sure those of you here who speak Hindi are a little bit unimpressed, okay? The rest of you don't actually hear a difference between the da and the da, okay? And by 12 months, typically developing babies actually can't make that distinction. It just sounds all like da to them. And then 10% of the time they hear ta, which is distinguished only on one feature, but we all hear the difference. Whether we're 6 months old, 12-month-old, or adults, we can hear the difference between da and ta. And so we're asking the question, do we see, the babies are listening to this, and what does their brain tell us the baby's able to perceive in terms of differences? Okay. So, what you see here on this side, to the left in blue are our low-risk controls, and we try as much as possible in our graphs, in our figures on the ISP, that low-risk controls are blue, high-risk are red. What you see here at the top, these are the 6, 9, and 12-month-old babies, okay? And what you see here is that the dashed lines are what we call the deviants. That's the deviant speech sounds, the da and the ta, look different from the standard at 6 months in both groups of infants, okay? They're still looking different from the standard at 12 months, at 9 months. By 12 months, what we're seeing is that the Hindi da is traveling now, is not significantly different from the standard da, and we see the same in both groups. So actually speech perception abilities are developing the same in both the high and the low risk infants. This is an early part of the ERP signal. This is at around 150 milliseconds. That's called the P150, but you can see there's a range. This is time along the axis here. Now, we then actually start to look at the late slow wave, the part of the signal that's a little bit beyond this piece. The reason we look at that is that that part of the wave is sensitive to asymmetry, okay? What you see here is that there's no difference between, one is the left hemisphere, one is the right hemisphere, okay? There's no difference at 6 months in either group. They're responding in the same way. By 9 months, we're seeing a significant difference in the response of the two hemispheres, okay, which continues at 12 months in the low-risk infants. But that's not seen in the high-risk infants. So they're not showing the same development of asymmetry for the functional organization for language in the first year of life. This is to show that at 12 months, these are our lowest controls, actually asymmetry correlates quite significantly with a parent-report measure of their language. So there's quite a close relationship between brain and behavior here. Those children who have greater asymmetry actually seem to understand more language at 12 months, okay? No such relationships. This is not a significant relationship at all. Another way of putting that is, yes, they're showing this atypical asymmetry. And what I forgot to say is, the data you saw are the same whether you put in the infants who have a positive outcome, an autism outcome, or whether you don't. And I think the data I showed you here are the ones who don't have an autism outcome. The point is, it doesn't matter in terms of behavior. It's not a marker of a problem because their language is not traveling with this asymmetry. You can have just as good language even if you're not showing this asymmetry if you're in this high-risk group, okay? We then followed this up at 18, 24, and 36 months. Instead of listening to speech, they're now listening to words, okay? Because, of course, by that point, children have already acquired a fair amount of language. And they listen to different sets of words. Some of the words they hear, we know that they clearly know because the parents tell us that. And then there's a group of words that they listen to that they have no idea what they mean, okay? And based on the work of Helen Neville and Deborah Mills, we know that between 14 and 18 months, so at the period prior to our first assessment, once there's a real developmental shift. Originally, both hemispheres begin to respond to words, and then there's a developmental shift where you see a greater, if you like, a greater signal on the left hemisphere. They show a greater asymmetry, more sort of advantageous. And so we took a look at this. They actually see a faster response in the left hemisphere based on the electrophysiology. We're looking at the latency in the N600, that's at 600 milliseconds after they listen to the word. And these are very preliminary data, which may or may not hold up as our sample increases. But what you're seeing here is that, in fact, and this is, I think, data only at 24 months, okay? What you see here is that for the low-risk controls, okay, there's no difference in their response to the the known and unknown words. These are the known words in solid, and these are the unknown words in the right hemisphere, but they're definitely showing a faster response in the left hemisphere, and that's basically replicating the Neville and Mills data. We're not seeing any such difference, okay, in either hemisphere. So in the high-risk infants. So it seems that this difference in this atypical lateralization for the functional organization of language continues. Okay. We then have taken a different kind of analysis to look at connectivity. Now, we know from a lot of work that the emerging idea that in autism, as well as other neurodevelopmental disorders, there's problems in anatomical connectivity, the fibers that connect, or the synchrony between responses across different regions, so that there are fewer long-range connections. We see more local activation, potentially more short-range, but I think that's fairly controversial still, okay? We know this, that in functional MRI, we see reduced functional connectivity between that green area on that slide I showed you on the orange area. There's several studies. And then using resting state EEG, people have seen differences in reductions in functional connectivity using that kind of a measure as well. So two people, a postdoc and I guess she was a postdoc when she did this work as well, Adrian Tierney and Julia Righi have been looking at this in our data. So what they've done is to look at connectivity, taking our speech EEG signal and then honing in on one part of the raw EEG signal. Well, it's not raw, it's been processed into the gamma band, which is quite sensitive in the higher frequency as a connectivity measure. And we're looking at this as a measure of event-related coherence between the leads in the frontal and the posterior region. We're looking at the strength of the connectivity or the synchrony between those two regions. But this is just to show you, again, this is quite preliminary data, and we're still playing around with exactly which parts of the E, which leads we should be investigating. But what you see here is this is developmental change between 6 and 12 months, okay? And what you see is that there's increased gamma coherence in the left hemisphere between 6 and 12 months in the response to the speech sound in the low-risk infants in both the left hemisphere and the right hemisphere. And what we're seeing is in the high-risk infants, no change in the left hemisphere, and in fact a somewhat decreased asynchrony, although that's actually not so, it's just that it's particularly significant at 12 months here. So this is another way of saying that we're still seeing using a different kind of measure, an atypical response to language. Now it seems we're focusing on the on this functional connection between frontal and posterior areas of the brain that are critical for responding to speech signals. We're then looking at, I'm switching gears now to a different paradigm and a different technology. We more recently introduced into our infancy program functional NIRS, and I'll talk a little bit about what NIRS, near infrared spectroscopy, is. It's another type of brain imaging method that is user-friendly, especially for babies because they don't have a lot of hair. And again, it's relatively cheap. It's really the very poor man's version of functional MRI. in a way. It's very poor in that way, but you do get good, relatively, if you just divide the brain into front, back, left, and right, well, it's a little bit better than that. Anyway, the paradigm that we're using, there's very little work in the normal developmental literature using NIRS, and we felt that it was important to take a paradigm that had been developed by others. What the babies are listening to, and you can see here we're doing this in the first year of life, 3, 6, 9, and 12 months, is they're listening to artificial words, and these either have a real statistic, a significant pattern to them, like penana, there's a repetition of the syllable within the word, okay? Or they're listening to words that have no repetition pattern in it. And this is like a precursor. I mean, you've got to pay attention to the statistical regularities and repetitions in what you hear, because that's how language that's how language is organized. And if you're not paying attention to those statistical regularities and irregularities, you're not going to really be acquiring language. So it's a good sort of precursor, plus it is in and of itself a linguistic stimulus that the child is listening to. Okay, so this is to show you what NIRS is. Like functional MRI, you're not measuring anything to do with brain activity itself, which is something that, in fact, you are able to do with EEG as an end point, but it is the electrical activity of the brain that you're picking up. You're not tapping anything that's actually the brain, you're tapping the blood flow like you do in fMRI. And basically we use light probes, okay, to detect blood flow, just like if I hold this, you see? The light from my laser is able to project it through to my finger. That's pretty much what you're doing here, except that it's not a laser probe, although I think some NIRS systems do use that. And you measure both the oxygenated and the deoxygenated blood, and you basically can separate out the signal between the two. And we have a little homemade cap. You put these probes on. They're not nearly as comfortable. So you try to minimize the number of probes that we put on the baby's caps. It's not very comfortable. But our babies, it turns out, is if you start babies on these things at 3 months, amazingly, when they come back at 6 months or they're sort of used to it. I mean, you'd think they'd only had it once for maybe 40 minutes, but it turns out those babies do much better than if you bring them in when they're, let's say, 9 months, they've never had this before, they don't like it as much, it's much harder to do. What Brandon Keene, a postdoc in our lab, has done has been to analyze the NIRS data, and in particular, to do a kind of a connectivity analysis of the data for this speech and for this speech paradigm where they're hearing these nonsense words with or without repetition patterns. And basically what you do is you massage the data like one always has to do, process it. It's not massage. Massage has a different connotation. Process the data, filter it, et cetera, and then we take 4 regions of interest, with 3 probes for the 4 regions. And the 4 regions are front, back, left, and right. Okay, and here's what you can see are the placement of the probes in the left and the right hemisphere as the baby's listening to these speech signals. And this is a little bit of a tricky slide to read. Here on the top are our low-risk infants, and this is very preliminary data based on small numbers. And here's the high-risk infants. And this is age 3, 6, 9, and 12. And the warmer the color, okay, the higher the correlation between the response in these regions. So it's a statistical correlational measure that we take as a proxy for connectivity, okay, which is pretty similar to the way people have done this kind of work in fMRI studies as well. And the warmer the color, the more the connectivity. The higher the correlations, the greater the connectivity. And basically what you can see, I mean, we don't have to go into the details because I know the pattern will change over time, but in the low-risk infants, we're seeing more warmer colors, these beautiful yellows and oranges. So it's basically saying that by 12 months, we're seeing much more in the way of connectivity, and this is both cross-hemisphere, the left front with the right front and the right posterior with the left posterior. The one remaining cool is with the left anterior with the right posterior, which we're not expecting much in the way of connectivity. In contrast, there's really not much change over time in the strength of the correlation. This is the one difference, the right anterior with the left posterior, which is a little bit anomalous. I don't think it's particularly interpretable, this red block here. And otherwise, it stays a lot cooler than it does. So we're seeing on this other measure, again, a little bit less in the way of connectivity. Switch now to our eye tracking measures, where we're using eye tracking in this study as a measure of language comprehension, okay? And in particular, we're interested in the speed of processing, okay? And built on work by Anne Fernald. And what Anne Fernald showed was during the second year of life, second to third year of life, infants are faster at processing language. That's not so surprising. And the way she does this is you present two pictures on the screen and you say a word. You know, look at the ball. And the most precise measure of processing speed that you can get is if when the child actually hears the word ball, they were actually looking at the other picture, which was a picture of a book. The speed with which they then switch, because now they've heard look at the ball, when they hear the word ball itself, how quickly do they switch to look at the other side? There's lots of other measures we could look at in these data and are doing. I'm just going to present that as a speed of processing measure. Why? Because what Anne Fernald has shown is that infants who are slower in their response time are at greater risk for SLI, specific language impairment, or language delay in the third and fourth year of life. Okay, it's actually quite an interesting predictive measure. So this is, the analyses have all been done by another one of my doctoral students, Maya Chita. And you can see, so this is just the trials, you know, they see things on the screen, and then the pictures remain on the screen, and they hear the word. At this point, after they've been able to look at the pictures, now they hear the word, and it stays up, and we're measuring the time course of their looking behavior. And these are the preliminary data that we've gotten at 18. We do this at 18, 24, and 36 months. And what you see here is this is, you know, time across here, and these are our low-risk controls, and these are our high-risk infants. And this is the amount of time after they've heard the word that they are able to switch to the other screen. Well, of course, initially they're just still looking, and these are only data when they've been looking at the wrong picture. And what you can see is among the low-risk controls, they get faster and faster between 18, 24, and 36 months. And that's a nice replication of what Fernald found. And what you see here is that at 18 months, the high-risk infants look like they're pretty good. They're looking like the low-risk infants. They show a little bit of advance at 24 months, but they're not showing any more increases in processing speed at 36 months. We're not quite sure what that means at this point. We still need to explore lots and lots of things, but the point of that dreadful slide I showed you early on about the complexity of this project is that it's a lot of work to get beyond this, and she just produced these graphs very, very recently. But even these high-risk infants, we know that they are at higher risk for language issues. And maybe processing speed is a little bit sensitive to that. And so there's something a little bit different about the way they're processing language. Their vocabulary scores are still in the normal range. But they're taking a little bit more time, and so that's somewhat of a liability, perhaps for greater problems with language, and that turns into reading issues when they're a little older. The final part I want to talk about additional contributions to language. And this is the work of my wonderful doctoral student, Megan Thompson, who those of you who have been here for a while may know. She was a graduate of UC Davis, and she has spent time here, and I want to thank you from the bottom of my heart for training her so well and sending her my way. Okay, so what we know is that infant gesture, communicative gesture, pointing behaviors, nodding and shaking your head, waving bye-bye, all those gestures, gesture predates language, and the more you gesture, and the greater variety of gesture, that's the best predictor of language development in all children, including children with ASD, and we and other groups have demonstrated that repeatedly. Gesture's the single best predictor of language. I think there's lots of reasons for it. It's not just communication, and it is, and it remains part of language. As you can see, I'm still gesturing. We don't give up gesture when we start to speak. But it also incorporates the motor components. There's somewhat of a learning piece to it, so there's an imitation component to it. But it also is primarily joint attention, expression of joint attention. So when I say it's the best predictor, I think it's because it incorporates multiple components. It's not to deny the significance of everything else. And what Susan Golden Meadow has shown is that maternal gesture, we've always known that the mother's language predicts their child, the rate of language development in their children. All typically developing children with no risk for other problems will learn to speak, but the more a mother speaks to their child, the richer their language is going to be and affects their time, the onset and rate. But what Susan Golden Meadow and Melissa Rowe have shown is that not only mother language, but actually the more mothers gesture to their children in that period, that first year of life and the early second year of life, is also an independent or over and above contributor to child language. So what Megan looked at was, well, to what extent does maternal gesture predict infant language in high-risk infants? So we coded infant gestures from the observational assessment, but also from a mother-child play session, and mother's gestures. So we coded the infant gesture and the mother's gesture at 12 months, and then we're using language outcome at 18 months. Okay, and that's taken from the Mullen, which is a standardized developmental assessment. So we're asking the question, does infant gesture predict later language, and does mother gesture predict later language? And this is just to say the kinds of things that we're coding, and these are pretty pictures. If I was adept at showing videos, I would show them, but I don't. So here's the data, again, blue low risk, red high risk. These are somewhat earlier, but we've definitely replicated this. Infant gesture at 12 months in the low risk infants is nicely predicting language at 18 months. And actually we see the same, although there's greater variance in the high risk infants. overall, and I don't have a slide to show this, we do find that when we look at the amount of gesture in the infants with an autism outcome, they show the lowest. So they'd be at the extreme low end of not producing much in the way of gestures at all. They definitely gesture less. Then we looked at maternal gesture at 12 months predicting to language. And in the initial analysis, we get a beautiful replication in our low-risk infants of the Golden Meadow data, but we actually didn't get a significant relationship here. So we then went back and asked the question, well, what's going on? Why aren't we seeing this? And so then what Megan went back and did was she said, well, you know, sometimes the babies aren't looking at their mothers when their mothers are gesturing. So maybe I ought to recode the data and just look at when the infants are in fact more or less looking at their mothers when they're gesturing. And then when she went back and did that, she found that there was a significant relationship. There's still a fair amount of variability. I think what's interesting about this is it didn't really change much for the low-risk controls when we did this, but it did for the high-risk. And that's, I think, a little bit suggestive about perhaps the high-risk infants have somewhat more difficulty that when they're busy looking at this and they're not actually looking at their mother, they're not really paying it, they're not able to process what the mother's doing out of the corner of their eye in quite the same way as the low-risk infants do. And that's, I think, something that's quite interesting. We then asked the question, and this is really a question, we really don't know the answer to this, well, overall, how much are the mothers gesturing? And what you see here is we separated out the kids with an autism outcome from those without. What's really interesting is that the mothers of these high-risk infants are actually gesturing more to their infants than the low-risk controls, mothers. Okay, so so they've learned. Now, keep in mind, they already have a child with autism. They've been through the mill. They know what they're doing, and we think this is really quite a provocative finding. Interestingly, there's enormous variation. We don't want to be saying, we know that overall these mothers are gesturing less. This, in our view, is driven more by the fact that these infants themselves, even at 12 months, are probably less responsive. And we're now trying to do some more fine-grained coding to get at that. But it's not that they're so low. They're actually, if anything, intermediate between these mothers and those mothers. And you can see there's a lot of variation. So just to summarize, I'm coming to an end. This is just to summarize what I said. There's a lot of heterogeneity in the language of children. We see atypical structural and functional organization in both people with autism and unaffected siblings, although the unaffected older sibling work has only been done on structure. We see this emerging very early in infancy, and I think that's interesting, important, and surprising, but it's emerging as an endophenotype that's not necessarily traveling with any behavioral deficits, okay? We even see not just atypical asymmetry, but some preliminary evidence for reduced connectivity in the siblings that may also be an endophenotype. And again, seeing this emerge fairly early. Whether this resolves as the children get older is something obviously that we need to be pursuing. There's slower processing speed by age 3, and that gesture, both infant and mother gesture, is a strong predictor of language, not just in preschoolers, which the earliest studies on this showed, or toddlers, as some of our studies and others have shown, but in fact going back to the infant period. What I want to say is, you know, it's just beautiful this afternoon sitting with Lori Vismara and looking at the parent training of the ESDM program, and you know, they've got it all there, and there's a separate unit. I mean, even though it's incorporated in everything else they're doing, but there's a separate unit on helping parents recognize the importance of gesture along with everything else in those for the early children. Future directions. Oh, we've got so much to do, guys, and just not enough time to do it. We want to look at the longitudinal growth trajectories in all our measures. We want to integrate our measures much more so than we have done. You could sort of walk away from here and say, here are all the gaps in Helen's talk. Looking at I've talked about all the language pieces. We're also seeing some atypical asymmetry patterns emerging in our high-risk infants in face processing. And I'm not going to talk about that, but the question is, well, do these travel together or not? Is there some relationship going on here? We think we're seeing a lot of things that are different in high-risk infants during the first year of life, none of which are really positive markers, if you will. So we're trying to pursue now some sort of an aggregate or cumulative risk model across our measures, and perhaps try to integrate these. And we've actually already started this with some work on this with people at Pat Levitt's lab in USC with some of the genetic profiles of our high-risk infants. So all I can say is thanks to you, and also to the people who have been funding our work all these years, my major collaborators in the studies I've mentioned here, all the staff and the students in my lab and at Children's Hospital. And here's a picture of our infant sibling group, which should be somewhat familiar to David. Picked this one of the many, but it's a great photograph. And then finally, of course, and I know I don't have to remind you, it's the wonderful children, our amazing, amazing families who have done so much to really let us look into their lives and learn so much about them, and inspire us to continue the work that we're doing. And thanks to you again.
The UC Davis Mind Institute was created in 1998 with a promise to find cures for neurodevelopmental disorders. Every day, our physicians and researchers come closer to fulfilling that promise. Their groundbreaking research on autism, Fragile X syndrome, chromosome 22q11.2 deletion syndrome, ADHD, and other brain disorders are helping children achieve their fullest potential. Please visit our website to find out more about current studies, upcoming events, and how you can help make a difference.